So I'm going to talk to you today about a tropical retrovirus which causes cancer of mature CD4 T cells. And this cancer is really devastating, but I'm going to hope to convince you that it's interesting mechanistically for immunologists because we can learn quite a lot about the biology of human T cells from studying it. So first of all, I'm going to give you a very brief background about the virus that causes this malignancy, and that's human T lymphotropic virus 1, or HTLV1, and it causes adult T cell leukemia lymphoma, or ATL. So the key research questions that I'm trying to address are, first of all, how does HTLV1 transform T cells, but also whether we can utilize the existing CTL responses in ATL patients to kill primary leukemia cells. So hopefully I'll sum that all up to a cohesive story. So HTLV1 is a retrovirus that is found in the blood. It's passed from mother to child where it establishes a lifelong infection. It's widely distributed across the globe, but we see high frequencies of infection in people in sub-Saharan Africa, South America, the Caribbean, and particularly in Japan. And has this strange clusters of high endemicity are found, which might be geographically very close to regions where there's very low prevalence of HTLV infection. And some examples of this are the fact that in Japan, where 1% of the population is infected with HTLV1, most of the viral burden is found in a small region in southern Japan, Kagoshima and Kumamoto. And similarly in Australia, most of the cases of HTLV1 are found in a very small region in northern Australia, in indigenous people, where the prevalence is up to 40% of people infected. So 95% of people who are infected with the virus are actually asymptomatic. However, 2 to 6% will develop this CD4 malignancy, adult T cell leukemia lymphoma, where they have a CD4 lymphocytosis, but also these strange cells with flower-shaped nuclei in the peripheral blood. We've known about ATL for 30 years, but unfortunately we haven't been able to treat it satisfactorily. Most cases don't respond to chemotherapy, and if you're diagnosed with some of the aggressive subtypes of ATL, you're likely to be dead within a year. So we have really clear motivation to try and improve outcomes for people who are infected with HTLV1 by investigating the oncogenic process to develop new therapies and diagnostics. So what do we know about the transformation process? There are two well-established factors that increase your risk of developing ATL. And the first is perinatal infection with HTLV1, and the second is poor control of the virus. And people with a high viral loads have a 20% risk of developing ATL. So ATL manifests usually around 53 years of age, in the UK cohorts anyway, and a recent molecular analysis has revealed that the malignant cells are clonal T cells, and they're also infected with HTLV1. Like other cancers, there are many mutations in the host genome, and a lot of these recurrent mutations actually activate or gain-of-function mutations which encourage increased signaling on T cell receptor pathways. So we also see mutation in the viruses that are carried in the malignant cells, whereby CTL epitopes in particular are silenced or deleted inside the malignant clone. Okay, so the key to understanding what's going on in this big black box that I've got here, which represents the transformation process, is trying to understand the biology of the virus itself. So HTLV1 preferentially infects memory T cells and is found at very high frequencies in cells that express this cellular protein, cellular adhesion marker 1, CADM1. Like other retroviruses, its RNA genome is reverse transcribed to DNA, after which a single copy of the viral genome is inserted into the host DNA. And we routinely measure this, the number of proviruses that are carried in peripheral blood for clinical monitoring of infection status. So HTLV has a kind of unusual mechanism of infectious spread, whereby it hijacks the host immune synapse machinery to generate these tight junctions between an infected cell and a target cell, across which virus particles are passed. And you can see here in a micrograph, the viral capsid protein is accumulating at the synapse between two cells during infection of a new target cell. So a really key feature of this is that the new copy of the viral genome is always integrated totally randomly into the host DNA. So this kind of effectively makes a molecular barcode on each new infected cell. So we can track clonal evolution over time using these markers. HTLV1 can also drive mitotic spread of infected cells through expression of viral proteins. And this establishes T-cell clones, which share common T-cell receptors, but also common integration site of the virus. So the question we first want to try and address is, how does HTLV1 transform T-cells? To begin this, we first asked whether the virus is latent or active in chronic infection. And a very superficial analysis looks like, maybe it's latent, because over 10 years, the proviral load is remarkably stable in most infected people that are asymptomatic carriers of the virus. However, when we looked a bit deeper, we could see that there was quite a high rate of proliferation of infected cells in carriers. So with up to 20% of HTLV1 effector memory cells proliferating, expressing Ki67 at any given time point. So actually, during chronic infection, we have frequent division of infected cells. But to maintain this stable equilibrium, there must also be a loss of for each cell division, which generates a new infected cell, another one must be lost. So we think that this is happening through CTL selection. CTL in HTLV1 infected people are very abundant, chronically activated, and when we take them ex vivo and co-culture them with CD4 T cells from the same person, we can see that they kill cells that express viral antigens. So this balance, so the CTL are contributing to this balance between host and virus, maintaining this stable equilibrium over years. So the next question we had is, what is happening at the clonal level? Are new clones arising all the time and being eliminated? So we developed two assays to try and measure this. Off the screen. So the first is the HTLV1 integration site assay, where we amplify junctions between the virus and the host genome and use next generation sequencing and bioinformatics to map and quantify HTLV1 integration sites. And we generate frequency distribution data like this, where we can see in this patient with ATL, 98% of their proviruses are inserted into chromosome 9 at this particular position. So the other assay that I use, or developed, is analyzes a TCR clonality by flow cytometry. So first we use our known marker of HTLV1 infection, CADM1, to zoom in on HTLV1 infected cells, and subsequently we quantify T cells which are expressing one of 24 different TCRV beta subunits by flow cytometry, and we also generate a frequency distribution here. And we can see this is the same patient that almost 100% of their HTLV1 infected T cells are expressing TCRV beta 5.1. So by comparing different patients, ATL and HTLV1 carriers, we can see that in ATL patients, a single integration site, this is the dark green here, is dominating the proviral load, whereas there are a small number of other integration sites detected, which represent polyclonal background of non-transformed HTLV1 infected cells. In the carriers, we can detect thousands of different integration sites that are present at low abundance. And we see a similar pattern with the flow cytometry data, but we only detect clonal expansions within the HTLV1 infected populations of cells. So mathematically, we can extrapolate this to say that of our 400 billion T cells in an adult human, there are 6 by 10 to the 4 unique HTLV1 infected clones existing within that population. But again, we wanted to know, how is this clonality evolving over time? So we compared the clonality in asymptomatic carriers in chronic infection at 2 time points separated by 10 years. And we can actually see that the clonal hierarchy is incredibly stable over time. So clones undergo frequent cell division to maintain the numbers that we see by combining our flow cytometry data with this. So actually it's a really key observation, and it introduces the idea that actually there's quite a high risk of introducing errors in the genome by replicative errors due to the increased turnover of those clones. So we have access to a cohort of patients which developed ATL at this time point here, where we had previous blood samples many years before they actually developed ATL, and we're currently looking at the acquisition of somatic mutations as the malignant clone increases in abundance from low frequency in the peripheral blood to completely dominating. So our next question is, given that we know that CTL are instrumental in determining the proviral load and control of HTLV infection in asymptomatic carriers, can CTL actually kill primary leukemia cells? And is there any kind of possibility that we could utilize new immunotherapies to use a person's CTL response to kill the malignant cells in ATL? There's relatively little data reported in the literature on the CTL response in ATL patients. However, we do know that in comparison with asymptomatic carriers of the virus, there's a very low frequency of virus-specific CTL in the blood. And as I mentioned, in the malignant clone, immunodominant CTL epitopes are often deleted or silenced. There's a generalized immunodeficiency in ATL patients, which is evidenced by a high frequency of opportunistic infections. So the first question we wanted to ask was whether the malignant cells could actually present antigens to CD8 T cells. So how we did this was by using the flow cytometry panel that I had previously optimized, whereby we could separate T cells in ATL cases into 3 different populations on the basis of the TCR-V beta subunit that they were expressing, and also this marker for viral infection. So we could separate out the ATL clone here from cells that were infected but probably polyclonal and not malignant, and cells that had a low proviral load. And by comparing just gross levels of MHC class 1 expression in these 3 populations, we didn't see any defect in surface expression of MHC class 1 in the tumour. As I described as well, the tumours tended to express high levels of the viral protein when incubated, or were completely negative, and this is consistent with what we expected. So we were able to stratify our patients based on whether they expressed viral epitopes or not. So the assay that I developed to quantify killing of ATL cells works as follows. We co-culture ex vivo PBMC, total CD4 T cells from ATL patients with different ratios of autologous CD8 T cells, and then perform flow cytometry to separate out these different subsets I just described, and perform absolute counts, but also phenotype the surviving cells after incubation, after co-incubation with the CD8s. So what did we find? As you'd expect in asymptomatic carriers, we could very easily detect efficient, strong CTL killing of virus-expressing cells. However, when we looked at killing of the malignant clone in ATL, we couldn't see any cells being killed. And actually what was quite interesting is that we couldn't see any CTL killing of virus-expressing cells that were not malignant in these same patients. So as expected, they have a clear kind of less efficient ability to recognize and kill virus-infected cells. So we've performed, I'm sorry, this got cut off. We cultured CD8 T cells from these HTL patients with polyclonal stimulation for 2 weeks, just to see if we could maybe enhance the frequencies of CTL that recognize the tumor or the virus. And we found that we could restore the ability to kill virus-expressing polyclonal infected cells, but only in a few cases could we actually manage to kill the malignant clone. And interestingly enough, these were the cases that were expressing the viral antigen tags. So we were able to ask the question whether malignant cells which express tags are selectively killed, and the answer was yes. Within a malignant clone, any cells that were expressing tags were killed, and cells that were not expressing virus proteins were not. So, to sum it all up, we're building a picture of transformation, which begins with a very first hit, which is exposure to HTLV1. We think very early in infection, the proviral load set point and this clone frequency distribution is established. And one of the key factors in determining this set point is the CTL efficiency of recognition of viral epitopes. And this is also associated with a class 1 genotype that an individual has. This is followed by years and years of accelerated cellular turnover in HTLV1 infected clones. And we get a natural selection of clones that have even tiny increases in fitness relative to the general population of T cells. And this ends up where we have a single dominant clone with multiple genomic abnormalities in ATL. Our assays of clonality actually can detect individuals that are developing ATL about a year before diagnosis, which is actually quite useful clinically because it gives people time to plan appropriate treatment for these kind of individuals. And our assays of CTL lysis of the tumour have revealed that actually in an ATL patient, the virus and tumour-specific CTL are actually quite inefficient. And it appears that the virus antigen, TAX, is an immunodominant CTL target, but we can clearly see that it could be quite easy for the tumour to evade this response. So we're currently investigating other approaches where we can use maybe cell therapies to clear leukemic cells in ATL. So that's all. Thank you very much. Most of this work was done in the laboratory of Charles Bangham. Nicola Chile did a lot of the initial sequencing experiments, but I've recently moved to the National Centre of Human Retrovirology, where we're trying to establish an independent group. So thank you very much for listening.